So thank you. So just to touch on a few housekeeping things, this is the third webinar for the quantitative genetics tools for mapping trait variation to mechanisms, therapeutics, and intervention that is sponsored by the NIDA Center of Excellence in Omics Systems Genetics and the Dome. And so today we're going to be talking about expression QTL and how we connect those to genes. So we sent out a survey a few weeks ago after our second webinar, and one of the really helpful suggestions that was given on that survey was to aim for a presentation that's close to an hour, and then add an extra 30 minutes for those that want to stick around and ask questions and have more of a discussion. So that's how we'll work it today. So feel free, if you've only got an hour, to log off at the end of the talk, or feel free to stay on and have a discussion with us to talk about in more detail some of the things that we're discussing in these webinars. I do need to make the apology of sending out the wrong link to you for those of you that had to register twice on my email. Lesson learned about copying and pasting. The slides are now available on GitHub, and the video and the slides will be available online at the OPAR website within the next couple of days. And we'll do another webinar here in a couple weeks, so we'll send out information all about that so that you can register and join in on that if you'd like again. So I think that about sums up the housekeeping items, except for that I will be sending out another quick survey, just 3 questions, in case you want to give us some feedback on the format or give us some feedback on other topics you'd like to see covered. So let's just jump right into it. I'm Laura Saba. I'm at the University of Colorado, and I'm co-director of the NIDA Center for Omics Systems Genetics and the Dome with Red, and I'm going to be talking today about expression QTL. So just to remind you what this whole webinar series is about, is we're trying to transverse this path in a forward genetics approach where we start off with a phenotype and a genotype, identify the QTL, and then identify genomic regions, genes, and pathways that could be responsible for that association between the DNA variant and our phenotype of interest. And so I wanted to start off by just recapping what we've already presented to get everybody on the same page and give you a reminder of the things we covered before. So if you remember, if you attended the first webinar, that was Shauna gave us a great introduction to quantitative trait loci analyses in general. So he was able to tell us what they are and teach us how, what genome scans are and how we find these QTLs through markers or DNA variants to find that link between DNA and our phenotype. Then for webinar 2, Rob built on this knowledge of QTLs and showed us how to do it within Gene Network. So he gave us a review of rodent models and human models looking at substance use disorder. He walked us through on Gene Network how we can take the phenotype data, do some initial preliminary glances at it, so looking at normalization and blocking and distribution, and then go ahead and do that actual mapping of the QTLs right on Gene Network. And then he ended the presentation by observing what genes were physically located underneath these QTLs that were identified. Sorry, I keep pushing the wrong button. So where I'm going to take over here is I'm going to talk about a slightly alternative pathway to identifying genes underneath a QTL and start talking about how we can incorporate RNA expression to link that genomic region or those QTLs with genes. And so we'll go over, the first half will be about expression QTLs for individual genes or individual transcripts. And then we'll talk about taking that a step further and looking at co-expression networks and how we can link co-expression networks to regions of the genome or to QTLs of particular phenotypes. So we'll start with an introduction to eQTLs. So like Rob mentioned in the gene network analysis, he looked at genes physically located in or And so a couple of reasons why we're interested in the RNA expression is it really is one of the first quantitative links between DNA sequence and phenotype. So DNA sequence differences there are usually yes, no, heterozygous, homozygous, where when we get to RNA, we really are talking about levels and slight differences that may have a large impact down the road. It's one of the first steps where DNA sequence and environment interact. And so we can see that right away with differences in RNA expression in different types of cells and different types of tissues. And RNA expression to that point can help us differentiate cell types and tissues within an individual. So incorporating this information into our analysis of how that causal DNA variant can affect our phenotype is also going to bring in knowledge about relevant cell types and relevant tissues. And finally, what we'll touch on at the end of this presentation is that also by using network analyses, we can, with these transcription levels and their natural relationships to each other, we can gain a little insight into genetic and environmental interactions. So when we start bringing RNA into this analysis of translating our QTLs into genes and mechanisms, we need to think of RNA as a mediator of the effect of DNA variants on a phenotype. So in our phenotypic QTL, we see this relationship, this dotted line between DNA and phenotype, and we really don't know what's going on in between. By adding this element of RNA expression, we can start to understand whether the DNA variant causes differences in RNA expression that in turn influence the phenotype. So this, by incorporating this concept of RNA acting as a mediator of that relationship between DNA and the phenotype, we can add to our information about translating that QTL. So instead of looking at genes physically located under the QTL, what we can do is look for RNA expression levels that are controlled by the same QTL that controls the phenotype. So when we start to think about some examples of how this would actually work in the cell, we bring back those effects that we talked about that were physically located. So again, a DNA variant in a transcription factor binding site or an untranslated region that affects levels. But we also incorporate information on what I'm calling in this context an indirect effect. So maybe the causal variant is in a transcription factor rather than in the transcription factor binding site. So the variants causing differences in the transcription factor, which then causes differences in expression of many other genes. And so one of our first assumptions, if we're going to bring in RNA into this model of how DNA affects the phenotype, is the assumption that our RNA expression is genetically controlled. And so from a very simplistic view, we think about heritability. So a lot of times we'll calculate heritability on phenotypes for humans and for rodent models. In the most simplistic case, in an inbred panel of animals, we can calculate heritability relatively easy. So we can look at the variation within an inbred strain, so within animals with the exact same genetic background, and compare that to the variability across strains. And this gives us an idea of how much of the variability in RNA expression is controlled by genetics or by differences in the DNA. So if you look on the left-hand side of this graphic, we see a toy demonstration of a really strongly heritable gene expression. So along my Y are expression levels, and then each one of my colors along the x-axis represent a different strain. So we can see that there's very little variation within a strain. So when they have the same genetic background, the RNA expression levels are fairly stable, but then we see relatively big differences when we look across strains. So when we do vary the genetic background, we get strong differences. Where in a non-heritable RNA transcript, we see a lot of variability within strain and just as much variability within strain that we do see across strains. So this RNA that has no heritability or very little heritability isn't a good candidate for the mediator of an RNA or of a DNA variant on our phenotype. And so we're looking for transcripts with this strong heritability characteristic. So when we go to map expression, we do it in a very similar way to what we did for the phenotypes that Shanak talked about a couple of webinars ago. So we simply map the RNA as a quantitative trait against our DNA variants. So again, we test for an association between each one of our markers or SNPs with our RNA expression levels. The big difference here really comes in to the computation of it all. So some of this is what we need to keep in mind when we're picking out tools and things like this to run these analyses. So now instead of running an analysis rather quickly, if we need to run 30,000 genes, then we have to be very intentional with our computational efficiency. And I just wanted to make a little note early on when eQTLs were first discussed, they were given the name genetical genomics. So we hear that often on here in more modern times, but originally we called it genetic genomics or genetical genomics, so studying the genetics that drive RNA expression. We can also classify these eQTLs into two different categories. So oftentimes we call them cis eQTL or trans eQTL, but local versus distal is a more accurate term for it. So in a cis eQTL or local eQTL, we're looking, the locus that controls transcription is near the physical location of the gene in the genome. So it's right in that promoter region, it's right in that untranslated region. It's something that has a direct effect on the gene of interest. Whereas a trans eQTL can be farther away on the chromosome or can be on a completely different chromosome than what the gene is physically located on. So if we come back to our example of a transcription factor, if there was a variant that was affecting the transcription level of a transcription factor on a different chromosome, but that transcription factor was necessary for the gene of interest to be expressed, that would end up looking like a trans eQTL. A couple of characteristics that we typically see on cis versus trans eQTLs. Cis eQTLs tend to be much stronger. So they tend to have lower p-values, higher statistical significance than our trans eQTLs. And we can rationalize that, based on these mechanistic effects that we've been thinking about and used to conceptualize eQTLs. So if the effect is more direct to the gene and doesn't have to act through an intermediate, then it's likely to have a stronger statistical effect than it would if it has to go through another transcript, another protein, or a more indirect route to controlling that expression. So with trans eQTLs, we also often see hotspots, is what it's called. And a hotspot is a eQTL that is in the same location for many genes that may not physically be located there. So in this graphic up here, we have a single element on a particular chromosome that actually controls multiple genes on multiple different chromosomes. We can also conceptualize it again, going back to my favorite example of a transcription factor. If the red square or red triangle here is a transcription factor physically located in its own cis eQTL, it may be creating a trans eQTL for many genes. So these blue circles represent genes that aren't physically located near the eQTL, but are controlled from that region. We talked a little bit about one of the benefits to using RNA expression to discover genes related to a phenotypic QTL is we can gain information about tissue and cell type. So this demonstration here is a graphics from the GTEx project that looked at cis and trans eQTLs across many human tissues. And a couple things that I wanted to point out about these graphics. So first of all, this upper triangle here represents the cis correlations across of eQTL effects across tissue, and the bottom triangle represents trans eQTL. So a couple of things that stand out fairly quickly on this graphic is that tissues that are similar tend to have similar eQTLs, both cis and trans. And so this box here represents different brain regions, and some of these other boxes along the axis represent slightly different tissues that are highly related. And again, you can see the brighter colors in these boxes. The other thing that we see is that brain regions tend to be similar amongst themselves, but tend to be rather different than the other tissues. And the final thing that this graphic alerts us to is that, as we expected, the cis eQTLs tend to be more conserved across tissues than the trans eQTLs. And you can see that by the brighter colors in the upper triangle than with the lighter colors in the lower triangles, and the difference in the scales on this. The other interesting thing we see about tissue specificity is when they went to compare cis eQTLs to look if they were shared across tissues. One of the things that was immediately apparent is that it was a very bimodal distribution when looking at the number of shared cis eQTLs. So cis eQTLs tend to be present in all tissues or most tissues, or present in a single tissue or just a couple tissues without very much in between. And they do tend to, and the final one is they do tend to have larger effect sizes, and the cis, then the cis have larger effect sizes and tend to be more consistent across tissues. So tools, we've been touching on this a little bit throughout, that the tools for mapping eQTLs are similar to any of the tools that can be used to map a behavioral or physiological QTL, but really efficiency is of utmost importance. So you can still use our QTL, and there's other tools such as QTL Reaper and Matrix eQTL. It's another R package that can do this rather quickly. And then when we're doing QTL analyses with population structure, there are several software out there that work to make this more efficient computationally so it can run these mixed models quickly. Because this does require a lot of computation, if you're thinking 30,000 transcripts across 10,000 variants, there have been a lot of databases for the rat eQTLs, and we'll talk particularly about rat because that's what our night assigner happens to be focused on. But both Phenogen and Gene Network have several rather large data sets of this eQTL data. Information. And so that's one of the benefits when incorporating it into these studies. So if you're measuring a phenotypic QTL, you can actually use eQTL information from different populations or different labs for linking it. So just to take a pause here and summarize about what we learned in this intro to eQTL. We know that expression QTLs are simply regions of the genome that influence RNA expression levels. So the genetically driven variation in RNA expression levels. When we separate them into local and distal eQTL, local eQTL tend to have bigger effect sizes and are more likely to be retained across tissues than trans. And eQTLs are mapped using similar methods and software. So now how do we link this information we've gotten from eQTLs to our phenotypic QTLs and eventually to genes? So what's our motivation? And so I'm going to call this a genetical genomics phenomics approach throughout, just to give it some kind of terminology. So when we use the first approach, when we only identify candidate genes that are physically located in a phenotypic QTL, we may miss genes that are controlled from that QTL, whose RNA expression is controlled from that QTL, but they don't physically reside there. So we're going to miss those genes with transit QTLs in the similar region. We also run the risk of identifying genes that aren't expressed in our tissue of interest. So for instance, if we're looking at a dependence-related trait for alcohol or drugs of abuse, we may not, and the gene may not be expressed in brain or a tissue that would be relevant to that disease, right? And so we could miss critical, and the third thing is we could miss critical information about how that causal variant influences the phenotype. But when we use this overlap of an eQTL with a phenotypic QTL, you're more likely to focus in on those genes from the tissues you believe are important. You're more likely to have a little bit more information about the mechanism on how that region influences the phenotype. And we might be able to identify broader biological and mechanistic themes by looking at genes that are controlled from the same region, although they may not all be physically located there. So they may have more of a mechanistic reason for having that shared transcription control. And we'll go into this a little bit more when we start to talk about networks. So in a genetical genomics phenomics approach, we're combining three sources of information. We're combining the sequence polymorphisms with the complex trait or diseases, so our phenotype here, with variation in transcription levels. And so in a very traditional genetical genomics phenomics approach, we look at each one of these comparisons, pairwise comparisons individually, and then look at convergence of results. So the sequence to the disease, we're going to use a QTL analysis. We can look at the correlation between a transcript and a disease. And then finally, we can calculate these eQTLs and make sure that the transcript is associated with the same polymorphisms that are associated with the disease. So again, we're looking for the convergence of all three of these analyses to a single gene to make it a candidate in a genetical genomics phenomics approach. And so just to give you an example of the way this has been done in the past, in this particular analysis, we were looking at alcohol consumption in a RI panel of rats. So we mapped the phenotype of interest. In this case, it's just amount of alcohol consumed. And then we took individual genes and first looked at their correlation with the phenotype and indicated that they had to be correlated with the phenotype. And then we looked to see that their eQTL location overlapped the bQTL location. And that's how we got our candidate list of genes. And then we're able to look at similar annotated biological functions among the genes and similar literature on these genes to get a better idea of genetic pathways involved. So the quick summary here is simply that by looking at the convergence of this information, we're getting more information about mechanism. We're getting candidate genes that we can link through both DNA and RNA to our trait. And so this genetical genomics approach, what we've showed you so far is looking at a gene by gene basis and then talking about a list of candidate genes in the end. But an alternative approach is to think of this in a network type setting. So why do we want to move from this single gene to a network type approach? First of all, it is logical to conclude that no gene product acts independently in the cell. That most, if not all, gene products interact with one another. we know that most tissues, especially brain, are complex hierarchical networks. And so we know that cell types, there's cell-to-cell communication, there's cell type to cell type communication, there's region to region communication, and all of these represent interactions and way genes work together. Another thing that comes up often, especially in substance use disorders, is that we can conceptualize many diseases as a failure in network regulation, is that some kind of network breaks down to produce the disease, to produce the phenotype. So it makes sense to model our data using that network structure as well. And then finally, it can provide more insight if we know the genes involved, if we know how they interact with one another, so that we can get a better idea of predisposition to disease and how diseases evolve. So there are typically lots of different methods for defining networks of genes or defining how genes and proteins interact. There's protein-protein interaction databases and networks that look both at experimental results and computationally derived results about how proteins interact with each other to form protein complexes and other mechanisms. There's annotated pathways and gene ontologies such as KEGG and the GO ontology that actually are derive these networks through annotation in the literature and other mechanisms and means. And then finally, one of the other methods, and again, this isn't an exhaustive list, is RNA co-expression. So instead of looking at relationships at the protein level or things that are derived from the literature, but actually looking at a mathematical or statistical mechanism for talking about the interaction between two genes or two RNA transcripts. And again, there's lots of different methods out there for doing that. Weighted gene co-expression network analysis is just one, and I'll talk a little bit about it more later just to be an example. But there's also very classic methods like k-means clustering and more advanced methods like Bayesian networks and Gaussian graphical models and more statistically advanced methods for identifying these co-expression networks. So I'm going to, the remainder of this talk, I'm just going to talk about the co-expression methods for identifying networks, and not that the other methods aren't valid or don't add a lot of information. It's just to focus the talk. So when we think of co-expression and using it as a mean of connection, the theory as a measure of connection, the theory behind it is that if expression levels of two transcripts correlate over multiple environments, then the two transcripts are likely to be involved in similar biological processes. So if gene A is increased under stress and B is also increased under stress, or A is reduced under hypoxia and so is B, if they tend to react in a similar manner across these multiple environments, the likelihood that they're doing similar things in the cell increases. So when we talk about genetic panels like the hybrid rat diversity panel or HS rats or other types of animals, we're talking about, instead of environments, thinking about stress and actual toxins and things like that, we're classifying these different environments as different genomes. So if gene A is high or low in a particular strain, then gene B tends to be low in that same strain. So in this toy example, the blue gene and the red gene tend to differ in a similar manner across all the strains. So we're hypothesizing that they have involved in similar processes. They may have a similar method of transcriptome control. Whereas the green gene here does not follow any of the similar patterns as the other genes. And so what do we gain by building these networks and identifying co-expression models instead of just considering each of the individual candidate genes? So we have found this in our research to be extremely helpful when we start looking at under-annotated or unannotated genes. And so this could be protein coding genes, this could be non-coding genes, this could be small RNAs that we may not have the KEGG information, we may not have the GO information. It may not create a protein that we could look at a protein-protein interaction with. But we can incorporate these genes into our co-expression modules and start to make hypotheses about what their biological function could be based on what the other genes it's highly correlated with do and what their known functions are. In a similar vein, many of the genes do lots of different things in the cell. And so when we can look for when genes have multiple roles, if we can look at the overlap of those roles between the different genes, that's when a network is associated with a particular phenotype, we may find out which one of those roles is important for this particular phenotype. When we start moving into, can we make functional hypotheses about mechanisms? Can we start thinking about therapeutic targets? When we have a network, our most significant gene in our association might not be druggable. It might not be the best one to target when thinking about therapeutics. But when we have that network of genes, we can start looking for alternatives or things that already have a drug or things that we know more about potential side effects with. And then finally, like knowing the biological function, we can also get information about cell types and regions, brain regions that may be of interest in a heterogeneous tissue. So if in our co-expression module, half of the genes are exclusive to dopaminergic cells, then those might be the cell type that's important for all genes within this module. So I'm just going to go into a little bit of detail about weighted gene co-expression network analysis. So this method has been around for a really long time. It's been used by many people. It's rather simplistic in a statistical point of view, but it's simple to use, and it's one that when I have trainees that are just beginning to learn about co-expression network analysis, it's a really good one to start with because they have great tutorials, they have an R package, and there's many people that are using it not only in the traditional way, but in new and novel ways. And so where WGCNA differs from simply looking at correlation is that we do some statistical and mathematical manipulations with, sorry, touching something on my mouse, statistical manipulations with traditional correlation coefficients to make our connections between our genes or our network resemble a scale-free network. And we'll talk a little bit about the motivation for wanting to do that in the next couple slides. And then the second thing that weighted gene WGCNA does above and beyond just a simple correlation is they also have created what's a robust measure of connection or co-expression between two genes by not only taking into account the direct correlation between the two, but also looking at the genes that are correlated with the other genes that are correlated with these two genes. So from a social network perspective, they're looking to see if these two genes, their friends are friends. So more evidence that this really is a robust association between two genes. Scale-free networks. So over here on the left-hand side is just a visual demonstration of the difference between what here on the farthest left is what we consider a random network versus what's on the right, which we consider a scale-free network. So just looking at the two pictures of the US, a lot of times we make the analogy of a highway system for the random network versus an airport system or an airline system for the connections on the right. So when we think about the highways or this random network, each node in the network has just a few lines or just a few links that are coming out of it. So each node is each dot. In our analogy, each city has about the same number of highways or roads that are coming into and out of it. So if we look at the distribution of the number of links per node, we see that most of our nodes or most of our cities in this example are have the same number of links, and we only see very few cities or very few nodes that don't have any links, and very few cities or very few nodes that have a lot of links. Whereas in our scale-free network up here, we see these red dots or these red nodes as kind of like hub cities for the airlines, where they have lots of nodes that are, lots of lines that are going in and out. And some of the other nodes up here have very few, maybe one or two that are coming in and out. And so the difference here in the distribution of the number of links per node in the scale-free is that most of our cities in this graphic only have one or two flights going in, and you just have a couple of cities that have a lot going in and out of it. And so that's a general description of what a scale-free network is. We have a couple of sources of information that led us to believe that RNA may work in a similar manner. We don't know that for sure, but one of the pieces of evidence that we have is that we can see some of these what are called small world properties in metabolic networks. So instead of metabolites being, the production of metabolites being done in a linear chain where one is needed to create two, which is needed to create three, and so forth, we see it, this arrangement, this linear arrangement can be really inefficient from getting from metabolite 1 to the production of metabolite 778. It takes a long time, it's very inefficient. If any one of these nodes breaks down or disappears, there's no way to get from 1 to 778. So it just isn't a very efficient biological model. Whereas if we think about hubs and think of it in a scale-free network type of paradigm, we can get quickly by moving from hub to hub from the beginning of the linear array to the end of the array, and that if we have any of the smaller nodes that fail in between, we're able to bypass them and still get the work done. The other source of evidence that I don't have a graphic here for is if we think about it in evolutionary terms of genes being added to the DNA and added to the system, so that older genes tend to have a lot more connections and tend to be your nodes rather than evolutionarily younger genes, which tend to have fewer connections. The other part that we talked about standing out and making WGCNA just a little bit different than correlation is this idea of indirect and direct correlations. And I realize that there's some ambiguity around the terminology here of indirect versus direct. So in the indirect, when we have high indirect correlations, if I'm looking at the association between the two gray genes or two gray nodes here, they have a strong correlation, but we also see that all of the green nodes, so all the other genes that are correlated, they're correlated with each other. So this green node is correlated with both of the gray ones and the other green one, that they're both correlated. So their friends are friends. Where if we look in this demonstration of a low indirect correlation, There's a high correlation between the two grays, but none of the other genes that they're correlated are correlated with the other gray node. So this tends to be a more robust correlation measurement, and this tends to be a less robust correlation measurement. So once we have our modules, so now we have, so we went from these measures of how to tell how connected two genes were. We use a modified hierarchical clustering method to then identify modules of co-expressed genes based on this new measure of connectivity or distance between two genes. But once we have the modules, one of the things that we need to be able to summarize the association of a module with a phenotype, or if we need to map the module, is to get a summary of the expression across the genes in a module. And so we're going to call this a module eigengene. And so these graphics here on the right are simply giving you a general idea of what I'm talking about. So if each one of these gray lines represents a gene across 18, gene's expression level across 18 samples, you can see it goes up and down, then the black line would represent the module eigengene for this particular gene. So it's trying to summarize the relationship among the samples across all the genes. And this is a similar graphic that you can see. This module expression heat map where we have genes along the Y and samples along the X, then is summarized into an eigengene expression value, where sample 3 tends to be high across all these genes and sample 6 tends to be low. We see that reflected in the eigengene expression. And so we typically use a PCA analysis to do that and look at the first principal components. And our end result, like this last graphic here, is eigengene expression, and we get one value for each one of our samples for a given module. And then just like we talked about expression, we can map this module eigengene using all the same tricks and tools and models that we had before. So to give you a general summary of what I wanted to cover with the co-expression networks is that by looking at genes as a group and talking about how they're related to one another, again, we're hoping to gain more information to develop those functional hypotheses down the road. Co-expression is one mathematical way for describing a network. And so this allows us to be more inclusive, but at times can be at the detriment of ignoring known information about interactions and assuming that there are things like specific relationships between types of relationships, whether it be linear or monotonic between two transcripts. We talked about WGCNA very briefly just because it's a popular method. It's a little bit easier to grasp and understand. And just like eQTLs, we can map our module eigengenes in that summary of those modules across the genome. So how do we link this all together? So again, all we've done is taken that genetical genomics phenomics approach and replaced an individual gene with a co-expression module. And so we've looked at genetic correlations, we've looked at QTLs, and we've looked for overlap with that module eigengene QTL. Just want to check how we're doing on time. I'm almost out, but I'm almost close. And so I just wanted to close with an example of when we've used this process of looking at networks and phenotypic QTL overlapping with module eigengene QTL. And so the particular example I'm going to talk about today we measured a couple alcohol metabolism related traits in an RI panel, and then used liver RNA expression to identify candidate genes. And so we did this very intentionally because we know a lot about what's going on in alcohol metabolism. So we had an expectation of what should come out of this procedure. So we did the typical analysis where we did our phenotypic QTL. So we were looking at alcohol clearance and circulating acetate levels, or acetate area under the curve in this particular analysis. We were able to identify several peaks you can see throughout here. We then identified our co-expression networks in our liver and looked for those that were associated with our phenotype. So these were associated with alcohol clearance, these were associated with acetate AUC. And one of the modules I want to point out here is this orange 3 that's associated with both acetate and alcohol clearance. When we overlap our maps of both the phenotypes and that orange 3 eigengene, so it's a particular module, we can see that we see a similar peak here for both the orange 3 eigengene and our alcohol clearance module. So indicating we do have that overlap. And when we look further into this orange 3 module, we see that the 2 most highly connected genes within this module are alcohol dehydrogenases. And so that's what we expected based on what we know about the process of alcohol metabolism. So in summary of the pQTL to the meQTL to network, we are gathering and looking again for convergence of information. We're using not only the identity of these genes, but how they relate to one another. And we were able to show that in a somewhat unsupervised approach that we get things that we would expect to get in this process. So I wanted to wrap up this portion, or the whole talk, I guess, by saying that by bringing in RNA expression, we're thinking about it being a natural mediator between DNA variants and behavioral, and that by incorporating RNA expression information, we're just broadening our possible mechanisms for how that QTL affects that phenotype, right? Because we would miss the differences in protein structure by only looking at RNA expression and eQTL overlap, but we would be missing some potentially critical genes by only looking at those physically located. So oftentimes it requires us to look at multiple sources of evidence for how to identify these genes. And oftentimes these co-expression networks, not only from a statistical point of view does it reduce some of that multiple testing burden, but it also allows us to bring in some of those genes that we just really don't have a lot of information on. And if we would have done it in a fashion where we only looked at a gene by gene basis, our natural human instinct would have been to ignore them completely and just go with the genes that we know. And so this is a way to kind of bring that information in and make sure that we're keeping those under-annotated or unannotated genes in our radar when thinking about what the next functional test is. The last one is just to remind you, the NIDA core who's been sponsoring all this, and we're here to help provide some training, help to find some study design and analysis services, and we have some pilot grants that we've given out to kind of build up these databases and resources. And finally, you know, I have a great group in my lab. We have some funding from NIAAA as well, as you saw by all those alcohol samples, to generate some of these data sets that hopefully can be used by many, many people. So I think I will stop my share there and see if we can attack some of the questions.
Thanks very much, Laura. There's some numbered questions in there. Joe had 2 towards the beginning. So if you just roll up the top of the chat. You can sort of follow along. Joe, if you want to unmute yourself and just ask your set of questions, go ahead. Let's see, do I have to unmute Joe, or can he do that himself?
Thank you.
There you are.
I have to go back and figure out what the first question was.
Oh,
So I see it in the chat, and the question was about splice
variants, right.
Right. So I think, I find splice variants especially intriguing. And I think our ability to capture and add splice variants to these type of analyses, whether there's some groups that have specifically looked at splicing eQTL or splicing QTLs, meaning genetic variants that could affect splicing, versus looking at eQTLs at the splice isoform level rather than at the gene level. So all of those things I think will continue to evolve, and I think are really easily incorporated in these kind of analyses. But calculating things at the isoform level requires really deep sequencing and a great depth of knowledge about what splice variants are out there. So it's requiring some more informatics at the front end for this type of analysis. And we as a rat community are working towards that. But all of those splice variants aren't annotated yet. And so that causes some ambiguity in some of the isoform level quantitations.
Do you have any sense of how much biology we're missing by not formally including splice variants in the test that we're doing? And second, I think that there's reasonable methods out there. There are methods out there to look at splice variants, and you referred to those that are annotated, but you could also do discovery of splice variants that could then be, if it's related to the trait of interest, then could motivate some secondary studies to validate the occurrence of the variant, and then you could do more that would again motivate further functional studies.
Right. No, and I totally agree. And we've done some work on this that the reason why I say that is because we have done some of the reconstruction with the RNA-seq. And so those are some of the methods that identify these novel splice variants. And those types of methods do really well at identifying splice junctions, but they do so at chaining them in the correct order. And so when we've compared that to things that we've gotten out of single molecule sequencing, right, which actually defines that the full transcript length, we see a few differences. So the reconstruction is better than the annotation, and the small molecule sequence is even better than that. And so that's why I hedge my bets a little bit there. But I do think it has a huge influence, with these data sets where we've used the single molecule sequencing, especially on brain, we're capturing 60,000 splice variants in brain, right? Where we have, 20 annotated, right? So we know that a lot of that's going on. So it's not only the splicing, but it's also those differences in 5' and 3' ends too, which could be really important. And I think that as we build these data sets and as these technologies improve, we can get more and more specific about the networks that we're building. So when we build them at the gene level, we're kind of grabbing at that low-hanging fruit, right? I don't think we're wrong in some of the things that we're identifying, but I think we're missing a lot of the more detailed and subtle differences like splicing and.
Right, because with expression levels, you're changing the amount of basically the same isoform. With splicing, you're potentially changing functionality of the encoded protein. Right.
And right now, most of we're trying to build these databases that are quantitating transcripts at that isoform level. But I think most of the results right now are at the gene.
Yeah. So my second question was related to something that you said later on about strain differences giving you a sense of a measure of heritability. But basically the strain difference is just a strain difference, is anything that differs between the strains, not only the nuclear genome, but mitochondria, the microbiome.
Mhm.
If there are parental behavioral influences, stress in the parents affecting stress in the kids, it doesn't. When we say heritability in the narrow sense that we mean, we're interested in genes because we're geneticists. But
Mhm.
heritability more generally is anything that's transmitted from one generation to the next in that nice linear strain-specific way.
Mhm.
And we need to take that into account when we look at those data in just the way that you said. To a geneticist, everything looks like a gene.
Right. No, and I agree, and I think that's going to be some of the, we're rederiving what we're calling the hybrid rat diversity panel, and so I think that will be interesting when thinking about some of these microbiome effects too, about where these strains were originally raised in different places, and if we resuscitate them in the same place and resuscitate them in the same environment, are we changing some of those microbiome effects, right? If they were environmentally driven and things.
Ron Kahn has a great paper, I think it's Cell Metabolism, maybe 3 or 4 years ago, where he looked at strains new from different sources, same strain, different sources, and then changes in the microbiome as they acclimated to the same facility. And there were remarkable changes in the microbiome, and they were correlated with changes in the host metabolic features.
Yeah.
It's a good point. It really comes back to Shaunak's first comment about method of differences. That was where we had the caveats of when that doesn't work too well. If you've got confounders you haven't taken care of. And that, of course, led to a lot of frustration on computing heritabilities. The nice thing about getting down to the QTL level is that at that point, you don't have to really concern yourself too much about the heritability if you have a strong linkage. But
yeah. What I was getting at too, though, is that if we see differences between strains, doesn't mean that it has a genetic basis. And there could be other features that could be just as interesting biologically, but outside of the nuclear genome.
To join the discussion now, just unmute yourself if you have any comments. It's not just the three of us here, so weigh in if you feel like weighing in.
Shall we go down the question list?
Yeah, I just want to make, we've got plenty of time, so we'll get to those, particularly since the next question is mine. So Laura, great introduction to cis and trans eQTLs. I asked a question, Carl gave an initial answer. Just want your thoughts. When we declare a cis eQTL or trans eQTL, the criteria are somewhat different because one is a genome-wide search initially, one is sort of a regional search around a gene, and that has some important implications for declaring that you found a cis or a trans eQTL. So talk about that a little bit.
So it's all how you apply it, right? Or the context of the experiment, right? Because one option is that we've kind of, in Colorado, we've kind of taken the approach before. It's we do genome-wide analysis, identify the top variant, and then declare whether the top variant is cis and trans, as opposed to separating it out into two separate analyses, right? Is one option for controlling a genome-wide rate. But there's also a lot of value to only looking for the cis eQTLs. But I do think that you can't really compare the threshold between the two, is my opinion, unless you're searching for them in the same manner.
Let's see, I'm just going through this. I think that was, that was question 3. And then you were about halfway through your talk. And Joe has a question, why are tests for cis eQTLs limited to local rather than genome-wide?
That related to what you were just saying, I think.
Why not genome-wide penalty for cis tests? You want to field that, Laura?
I think that was kind of the The approach I was talking about, if I'm interpreting in the question right, is that you penalize everything for a genome-wide search and then classify your hits as either cis or trans, rather than testing directly for a cis effect and only looking at cis genes,
So
trusting directly for trans.
do cis effects get a multiple testing penalty?
So it depends on how the QTL is applied.
Yeah.
My philosophy is a little bit more liberal. So, and it's based upon sort of I think a reasonable Bayesian prior, that if any variant is going to control gene expression, it's going to be a local variant. So that, self-control is sort of a prior expectation. And from that prior expectation, you actually expect in a sufficiently polymorphic population, every transcript to be a cis eQTL at some level. It may not be true in a population that has a lot of regions that are IBD, but if you imagine a hypothetical population where every gene is highly polymorphic, then every gene should be a cis eQTL, frankly. So from that point of view, the initial barrier for declaring a cis eQTL, in my opinion, should be quite should not involve any kind of genome-wide correction. It might involve a correction for the length of segment that's in LD with the gene. In a human GWAS, that'll be pretty small. That'll be 100 kb or less. But in that 100 kb, you might end up doing 20 to 100 tests. So in that case, you would need an FDR correction for all the local tests you've done. So you couldn't just blithely take a p-value of 0.05 as sufficient evidence of linkage. But it really, I think it depends a lot on your priors. And my prior is, again, that cis eQTLs are frankly no-brainers. And I find them interesting, but hardly surprising.
So Rob, your point is completely consistent with Carl's suggestion in that his suggestion of applying the FDR across all the cis QTL is exactly what you're talking about, except that the FDR has a empirical based interpretation. So it is estimating the prior in effect from the data itself, because you can kind of based on the shape of the distribution of p-values, you can estimate what fraction of cis eQTL are actually null versus not null. So his suggestion is completely consistent with your intuitive idea of how they should be treated. But I think in some ways applying the FDR has the advantage of taking out to some extent individual opinions, and you have a more formalized procedure. Anyway.
Great. Carl, you have anything you want to add? You're still on mute. I've tried to unmute you, but maybe you're not right in front of your monitor. We had a question from Herodotus. Where is that question?
I see it, Rob. It says, how to appropriately test for gene overlap between two modules. They build one with express data and everyone seems to compare. So this is a really really good question that we have been struggling with, or not struggling with, but investigating, let's say. It's a more positive way to put it. In some of the research that we've been doing computationally in the lab. It's really to compare two networks to say that a network has fallen apart or a network has been activated under different conditions. So there are some tools from example from WGCNA where they're looking at the robustness of a module. So what they would do would take the genes from E turquoise and look to see what their relationships are in the splicing data set. So then they can assign a quantitative value to how well that E turquoise and is conserved in the splicing data. But it's not necessarily meant to say, I've identified these modules independently, how much do they overlap? And so there are several, statistical tools to say, does this group of genes in the E turquoise module have the same relationships when I look at them in a different data set? But not as much as for the overlap. In a real, because an overly simplistic approach would be to simply look for enrichment of genes from the turquoise and the S turquoise. But what you're missing there is the weightedness based on how connected those genes are in the module. We've anecdotally found, as we've ran these types of analyses on different technologies and with different normalizations and things like that, that our modules tend to be really robust among those genes that are highly connected. So look at that module for liver, the alcohol dehydrogenases and those that were highly connected tend to be really robust no matter what we do to the data set. It's the ones that were there on the periphery that weren't that connected with the other genes that kind of fall in and fall out. And so strict, Fisher's exact test for the two models would miss that information, the prioritizing of that information. But I can definitely put a link in the chat discussion that we post on GitHub for the article from at least the WGCNA group that has this Z-score for replicability of a module.
Right, there's a question that just came in from Priscilla. You might have a look at that to the last. Has anybody compared cis eQTLs to hub genes in WGCNA?
So we've just done it anecdotally. We have a systematic view, but it would make sense that those that are most highly connected, especially for those modules that have a strong eigengene QTL, that it would be a cis eQTL at the hub. And so in some of our work where we've looked at the alcohol consumption in brain, our most highly connected genes in the module are ones that are physically located in that area and have a cis eQTL. And that sort of leads to, I think it was your question, Rob, about how to avoid LD artifacts.
Yeah.
Right? Implementation of WGCNA, we can't, LD could be one of the reasons why the genes are coordinated. One of the things that
some people may know exactly what that means.
So LD is linkage disequilibrium. And so it's saying that gene A and gene B their expression is correlated simply because they're physically located near each other on the genome. So during recombination, they tend to travel together, but the actual variant that's controlling expression for those two genes are two different variants, right? They just happen to always travel together during recombination. So they appear correlated, but it's just because they're so close to one another on the genome. Do you agree with that definition, Rob?
Absolutely. Yeah. How could I disagree?
And some of the things that we've done in a post hoc manner with our co-expression modules is then to look at partial correlations among genes within the module once you control for that QTL, for that module eigengene QTL. So if two genes are highly correlated and then we do a partial, and they're physically next to each other, and we do a partial correlation where we account for that QTL, and they're no longer correlated in that partial correlation, then it's a signal to us that, or it's evidence for us that those two were just correlated because of linkage disequilibrium rather than some mechanistic relationship between the two. And so again, I've got a whole slide on that. We can do a whole webinar on it sometime about what the rationale is behind that and how the statistics behind it work. But we've just done it simply from in a post hoc vein. I think I've seen other people do it where they adjust out a cis QTL effect of all genes and then do a WGCNA, which is another option on how to control for that.
I'm pleased to see that we have Jake Lucis on taking the class with us. He could be giving this, having coined the term eQTL in some papers in the early 2000s. Jake, if you want to unmute yourself and say hi, that would be great.
Hello, everybody. Good to see everybody.
So,
I've enjoyed this, Rich. Great talk.
Don't know, Jake, it was Eric Schatz's mentor. And so all of those very early corn, human, mouse, eQTL studies. And of course, he's been very busy in the field for the last 20 years since then. So good to joining us.
Thank you.
And if you want to give one of these courses, just let me, Laura, and Shaunak know.
Cool.
Trying to think.
There was one more from a little Herodotus, but maybe that got answered. Module size. What's module size?
So it depends. One of the clinicians in our department laughs because she says that that's the standard statistician and answer is it depends. When WGCNA, the creators of WGCNA, Steve Horvath and Peter Langerfelder, they suggested 30 genes being minimum module size. And some of their rationale for that is because if you want to do any kind of enrichment analysis afterwards, so if you want to look for enrichment of GO terms or enrichment of KG pathways, you need at least 30 genes to do that in any kind of reasonable way. In our lab, we tend to drop it down to 5. And the reason why we drop it down to 5 is because we know that of the genes that we're able to share with our technology, we might not expect really a strong correlation between a ton of genes. And so willing to do the knowledge-based analyses of looking it up and doing things like that. And to us, a smaller number of genes is much more manageable for looking up each individually in the literature to make a story. And we've tested it out with WGCNA. What happens is when a gene does not fit into a module, they put it into a clump that they call gray. And so that just means that it wasn't in a module that was over 30. When we adjust that minimum module size in our actual development of these modules, what we see is that gray tends to get smaller if we allow those smaller modules. So we're willing to take the hit on the multiple testing correction for more modules to be able to dig down a little deeper on some of these small ones. So it really depends on what you want to do next. And a lot of times it may even depend on what the phenotype you're interested in is. Is it phenotype that you expect 1,000 genes to be associated with or influencing, or is it a phenotype that you expect a smaller number of relevant genes in a smaller network to be involved?
You probably see that question from Shaunak at the very bottom.
How do your methods change for other omics data, such as microbiome, metabolism, etc., versus the transcriptome? So that's a good question. So, one of the things that we've been struggling with, again, struggling is the wrong word, let's be positive, which we've been investigating a little more, is once you start in my mind, the philosophy of the scale-free network is easy to comprehend at the protein coding gene level. When we start adding in things like non-coding or we're talking microbiome, might not expect that scale-free network distribution. We might not expect the same relationships between a non-coding and a protein coding RNA we would between protein coding RNAs. And so it's that piece of the WGCNA that I'm more hesitant with when we switch to other omics, is this idea of the scale-free network and whether or not that's something we want to enforce.
One other obvious difference is that some of the omes are rooted in the genome. The epigenome is, the transcriptome is, and the proteome is. In other words, the concept of having a cis and a trans eQTL makes sense for at least the proteome and the transcriptome. They don't for the metabolome, and they don't for the metagenome, and they don't for a lot of omes that are new. So in some sense, they don't have that built-in routing. So the lovely thing about the cis eQTLs is they are sort of a built-in standard for quality control and other, basically data quality control. Obviously, there's a lot of real biology there. So I had a question for you, Laura, about WGCNA. So it seems like when the scale-free, the exponent beta that's used to raise the correlation coefficients of, up to 8, or which is kind of the recommended default, that tends to kill all the negative correlates, which typically have lower correlation coefficients than do the positive correlations. So if you look at a distribution of correlation coefficients, they tend to be positively skewed. And now raise them to some power like 8, the negative correlations get washed out. But those negative correlations are really important. In your talk, you mentioned that you're defining friends of transcripts, but I'm often more interested in defining antagonists. And I find for WGCNA, I almost always have to rummage around and rescue the negative covariates. I'm just wondering whether you have a strategy for
So that's a good question. And I've never looked at in depth about the bias in the distribution of the negative to the positive correlations. There is options in WGCNA, and there is differences in philosophy on whether you actually do what they considered a signed network versus an unsigned network. So in a signed network, you're only correlating, you're forcing all of the genes within the network to have a positive correlation with each other, as opposed to allowing negative and positive correlations within the same network. I've always been a big fan of letting them both in. I think that there's not much value to only looking at one direction, because, so many of our pathways have both, like you said, protagonists and antagonists, and, we don't know that that's important to have that information.
Was that ever
together.
flagged? So in your Orange 3 module, could you tell me which ones were positive and which ones were negative?
Yes, I can.
Okay. I just wanted to make sure that data was actually preserved. I haven't followed that, but I really are sensitive to the imbalance. It's way more acute in human data sets. So if you look at GTEx, it's extreme. You can look at the top 500 covariates of a transcript, and every one of them will be positive. In rodent expression data, we rarely see that. It tends to be more balanced. But even in the rodent data sets, the positive covariates are more common, and it kind of makes sense because any of your batch effects are going to add on the positive side, almost always.
Right. Yeah,
So
that would be my interpretation of the overabundance of positive correlations.
I wish there were a way to take the negative. I mean, there is like a kludgy way. You take the negative covariates, give them a little boost in the negative direction so that they were perfectly balanced with the positives, and then raise everybody to the power of 8.
So I think the one last question, given we only have 5 more minutes, is what constitutes, this was from Joe Nadu, and it was, what constitutes to independently validate an expression module? And again, this is a really hard question to answer. And so I'll go back to so WGCNA, they have a method, and it's published in a paper. It's like, is my module replicable, right? And so they actually take, I believe, 9 different measures of replication. So is it the same genes? Is it the same order? Is the most connected one always the most connected one? Those sorts of things. So they have there's different aspects of the module that you can quantitatively measure to say that it's been reproduced. And so they combine that into a Z-score to do it. But there isn't a globally acceptable way to say that this entire expression module is validated, right? And so, like we talked a little bit about anecdotally, that we tend to validate the highly connected ones, and the low connected ones tend to not validate. Well, do you consider that whole module validated or not? And so there's a lot of, it's not an easy answer. We tend to use that overall, almost like a meta-analysis of different aspects to look at the robustness of our networks. So we'll take our WGCNA module, and then we'll do a bootstrap sample and see what that conservation of that module is in the bootstrap sample, and see how consistent that is when we do things like bootstrap or natural ways to
Feel free, anybody else, unmute yourself, and comments, questions, suggestions for improvement, all welcome at this point. Countdown, 10, 9, 8. All right, well, I guess we're heading into the weekend. Actually, I and Shauna heading into our next meeting.
Well, thank you guys for coming. Please expect a survey soon, and then we'll have all of this up on, and I'll give more detailed responses to the questions as well with references and such.
All right, Shauna, you can terminate recording, I guess.